Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In the vast tapestry of life on Earth, few creatures challenge our understanding of biological limits, like the microscopic tardigrade. These eight-legged microanimals, measuring barely half a millimeter in length, have earned the nickname water bears for their lumbering gait and rotund appearance. Yet beneath their seemingly innocent exterior lies one of nature's most extraordinary survival mechanisms, a biological resilience that defies conventional understanding of what life can. Tardigrades represent a fundamental shift in how scientists conceptualize the boundaries of existence. Discovered in 1773 by German zoologist Johann August Ephraim Goethe, these remarkable organisms have since revealed capabilities that read like science fiction. They can survive temperatures ranging from near absolute zero at -273 degrees Celsius to scalding 150 degrees Celsius. They withstand radiation doses 6,000 times greater than what would kill a human. Perhaps most remarkably, they can survive the vacuum of space itself. The secret to their extraordinary resilience lies in a process called cryptobiosis, literally meaning hidden life. When faced with extreme conditions, tardigrades undergo anhydrobiosis, expelling up to 99% of their body water and entering a state resembling death. Their metabolism drops to an unmeasurable level, cellular processes halt, and they curl into a compact structure called a ton. In this dormant state, they can survive for decades, possibly centuries, waiting for favorable conditions to return. Recent research has unveiled the molecular mechanisms behind this incredible feat. Scientists at the University of North Carolina discovered that tardigrades produce unique proteins called tardigrade-specific intrinsically disordered proteins, or TDPS. These proteins form protective glass-like structures around cellular components during desiccation, preventing damage that would normally prove fatal. Additionally, tardigrades possess an exceptional DNA repair system, capable of fixing extensive genetic damage caused by radiation or extreme environmental stress. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free, and it helps us grow this community together. The implications of tardigrade research extend far beyond basic biology. NASA has conducted experiments aboard the International Space Station, exposing tardigrades to the harsh conditions of space to understand how life might survive interplanetary travel. The European Space Agency's TARDIS experiment, Tardigrades in Space, demonstrated that these creatures can indeed survive and even reproduce after exposure to solar radiation and cosmic rays in low Earth orbit. Understanding tardigrade resilience offers insights into astrobiology and the search for life beyond Earth. If organisms on our planet can survive such extreme conditions, what might be possible elsewhere in the universe? The study of extremophiles like tardigrades is reshaping our conception of habitable zones and expanding the parameters scientists use when searching for extraterrestrial life. Tardigrades also hold promise for advancing human technology and medicine. Researchers are investigating how TDP proteins might be engineered to protect other organisms or even preserve human cells and tissues. The potential applications range from improving organ transplantation to developing new preservation techniques for space travel. As we stand on the threshold of a new era in space exploration, with missions planned to Mars and beyond, understanding life's extremes becomes increasingly crucial. Tardigrades serve as living proof that the boundaries of life are far more flexible than previously imagined. They represent not just remarkable biological curiosities, but potential keys to unlocking the secrets of survival in the cosmos. The story of tardigrades is ultimately a story about life's incredible adaptability and the power of scientific inquiry to reveal nature's most profound secrets. Deep beneath Yellowstone National Park's colorful hot springs and in the scalding depths of oceanic hydrothermal vents, life thrives in conditions that would instantly kill most organisms. These environments, where temperatures soar beyond the boiling point of water, harbor some of Earth's most extraordinary inhabitants, thermophilic organisms that have revolutionized our understanding of life's thermal limits. The discovery of thermophiles represents one of biology's most significant paradigm shifts. Until the 1960s, scientists believed that life could not survive above 70 degrees Celsius. This assumption was shattered when microbiologist Thomas Brock isolated Thermus aquaticus from the hot springs of Yellowstone in 1966. This bacterium thrived at temperatures exceeding 70 degrees Celsius, opening an entirely new frontier in biological research. Thermophiles are classified into distinct categories based on their optimal growth temperatures. Moderate thermophiles flourish between 50 and 60 degrees Celsius, while extreme thermophiles or hyperthermophiles prefer temperatures above 80 degrees Celsius. The current record holder, strain 121 isolated from hydrothermal vents, can reproduce at temperatures reaching 121 degrees Celsius, well above water's boiling point at sea level. The biochemical adaptations enabling thermophilic survival represent evolutionary masterpieces of molecular engineering. These organisms have evolved specialized proteins with enhanced thermal stability achieved through increased disulfide bonds, modified amino acid compositions, and altered protein folding patterns. Their cell membranes contain unique lipids with branched-chain fatty acids and ether linkages, instead of the ester bonds found in mesophilic organisms. These modifications prevent membrane fluidity at high temperatures, maintaining cellular integrity. Thermophile genetics reveal additional, fascinating adaptations. Their DNA contains higher concentrations of guanine and cytosine nucleotides, which form 3 hydrogen bonds compared to the 2 bonds in adenine-thymine pairs. This increased bonding strength prevents DNA denaturation at elevated temperatures. Furthermore, thermophiles produce specialized heat shock proteins and chaperones that help refold damaged proteins and maintain cellular function under thermal stress. The discovery of thermophilic archaea, particularly members of the genus Pyrococcus, has been particularly significant. These ancient microorganisms, whose name literally means fire sphere, represent some of Earth's earliest life forms. They inhabit deep-sea hydrothermal vents where temperatures exceed 100 degrees Celsius and pressures reach hundreds of atmospheres. Their existence suggests that life may have originated in high-temperature environments, challenging traditional theories about early Earth conditions. Hydrothermal vent ecosystems showcase thermophile diversity and ecological importance. These underwater oases support complex food webs based entirely on chemosynthesis rather than photosynthesis. Thermophilic bacteria convert hydrogen sulfide and other chemicals into energy, forming the foundation of ecosystems that include tube worms, crabs, and fish. The discovery that life could thrive independently of sunlight fundamentally altered concepts of where life might exist in the universe. Industrial applications of thermophiles have transformed biotechnology. The heat-stable enzyme TAQ polymerase, derived from Thermus aquaticus, revolutionized molecular biology by enabling the polymerase chain reaction, or PCR, technique. This breakthrough allows rapid DNA amplification and has applications ranging from medical diagnostics to forensic analysis. Other thermophilic enzymes are used in industrial processes requiring high temperatures, including paper manufacturing, textile processing, and biofuel production. Recent research has identified thermophiles in previously unexpected environments, including deep subsurface rocks and geothermal systems worldwide. Scientists estimate that thermophilic biomass in Earth's deep biosphere may equal or exceed all surface life, suggesting these organisms play crucial roles in global biogeochemical cycles. Understanding thermophile metabolism provides insights into carbon cycling, mineral formation, and the limits of Earth's habitable zone. The extraordinary survival capabilities of extremophiles like tardigrades and thermophiles emerge from sophisticated molecular machinery that operates at the cellular level. These organisms have evolved intricate biochemical systems that fundamentally challenge conventional understanding of biological limitations, revealing how life can persist under conditions previously thought impossible. At the heart of tardigrade resilience lies their unique approach to cellular protection during desiccation. When facing dehydration, tardigrades initiate a complex molecular cascade, beginning with the production of tardigrade-specific, intrinsically disordered proteins. These TDPs lack fixed three-dimensional structures, allowing them to form flexible, glass-like matrices around essential cellular components. The process resembles vitrification, where rapid cooling creates amorphous solids that preserve cellular architecture without forming damaging ice crystals. The molecular composition of TDPs reveals remarkable evolutionary innovation. These proteins contain high concentrations of hydrophilic amino acids that interact with water molecules, facilitating controlled dehydration. During anhydrobiosis, TDPs replace water in maintaining protein structure and preventing cellular collapse. Advanced spectroscopic analysis has shown that these proteins create protective scaffolds around organelles, DNA, and essential enzymes, essentially freeze-drying the cell while maintaining structural integrity. Tardigrades also possess exceptional DNA repair mechanisms that surpass those found in any other known organism. When subjected to radiation or extreme desiccation, their genetic material can fragment into hundreds of pieces. Upon rehydration, specialized repair enzymes rapidly reconstruct the genome with extraordinary fidelity. This process involves homologous recombination, non-homologous end joining, and unique repair pathways that scientists are still working to understand fully. Thermophilic organisms employ fundamentally different molecular strategies for extreme survival. Their proteins undergo structural modifications that maintain functionality at temperatures that would denature most biological molecules. Hyperthermophile proteins contain increased numbers of disulfide bonds, which act as molecular bridges, stabilizing protein architecture. These covalent connections prevent thermal unfolding and maintain enzymatic activity at temperatures exceeding 100 degrees Celsius. The amino acid composition of thermophilic proteins reveals additional adaptations. These organisms favor amino acids with charged side chains such as glutamate and aspartate, which form stabilizing ionic interactions. They also incorporate unusual amino acids and modified bases that enhance thermal stability. Some hyperthermophiles produce proteins with intrinsic metal cofactors that provide additional structural support under extreme conditions. Thermophile membrane biology represents another marvel of molecular adaptation. Their cell membranes contain archaeal lipids with branched isoprenoid chains linked by ether bonds, rather than the ester linkages found in bacterial and eukaryotic membranes. These ether bonds resist hydrolysis at high temperatures, preventing membrane degradation. Some species produce tetraether lipids that span the entire membrane, creating monolayer structures of exceptional stability. The genetic machinery of thermophiles demonstrates remarkable heat tolerance. Their DNA polymerases, the enzymes, responsible for DNA replication, maintain activity at temperatures that inactivate mesophilic counterparts. These enzymes have been extensively studied and modified for biotechnological applications. The three-dimensional structure of thermophilic DNA polymerases reveals additional disulfide bonds and modified active sites that confer thermal stability. RNA metabolism in thermophiles showcases further molecular innovations. Their ribosomal RNA contains modified nucleotides and additional secondary structures that prevent thermal denaturation. Transfer RNAs feature unusual base modifications that stabilize their L-shaped structures at high temperatures. These modifications ensure continued protein synthesis under conditions that would halt cellular processes in mesophilic organisms. Recent advances in structural biology have revealed how thermophilic chaperone proteins assist in maintaining cellular function. These molecular chaperones, including specialized heat shock proteins, help refold thermally damaged proteins and prevent aggregation. Some thermophilic chaperones form large cage-like structures that provide protected environments for protein refolding, functioning as cellular repair workshops. Comparative analysis of extremophile molecular machinery reveals both convergent and divergent evolutionary strategies for surviving harsh conditions. Extreme environments function as natural laboratories where evolution conducts its most ambitious experiments, pushing life into territories that challenge fundamental assumptions about biological possibility. These harsh frontiers, from the superheated depths of oceanic trenches to the radiation-soaked vacuum of space, serve as testing grounds where only the most innovative molecular solutions survive, driving evolutionary breakthroughs that expand our understanding of life's potential. Deep-sea hydrothermal vents represent perhaps the most alien environments on Earth, yet they teem with life forms that have revolutionized evolutionary biology. These underwater geysers, discovered only in 1977, create conditions resembling those found on other planetary bodies. Water temperatures exceed 400 degrees Celsius near vent openings, while pressures reach crushing levels hundreds of times greater than at sea level. Chemical compositions include toxic hydrogen sulfide concentrations that would prove lethal to most surface organisms. Within these extreme conditions, entire ecosystems have evolved independently of solar energy, fundamentally challenging terrestrial biology concepts. Giant tube worms belonging to the species Riftia pachyptila grow up to 8 feet long despite lacking digestive systems. Instead, they host symbiotic bacteria within specialized organs called trophosome that convert chemicals into usable energy through chemosynthesis. This biological partnership represents one of evolution's most remarkable innovations, demonstrating how life can transcend traditional energy limitations. Pompeii worms, scientifically known as Alvinella pompeiana, inhabit the most extreme thermal gradients on Earth. These polychaete worms construct tubes near vent openings, where their tail ends experience temperatures around 80 degrees Celsius, while their heads remain in relatively cooler 40-degree water. Their survival depends on unique bacterial mats covering their backs, which likely provide both thermal protection and nutritional benefits through symbiotic relationships. The evolutionary pressure cooker of hydrothermal vents has produced organisms with extraordinary biochemical adaptations. Vent bacteria have evolved enzymes that function optimally at temperatures that would destroy conventional proteins. Some species, like Pyrococcus furiosus, thrive at temperatures exceeding 100 degrees Celsius and actually require these extreme conditions for reproduction. Their existence suggests that life's origins may have occurred in high-temperature environments rather than the moderate conditions previously assumed. Antarctic dry valleys provide another evolutionary laboratory where life confronts extreme desiccation, temperature fluctuations, and intense solar radiation. These valleys, considered among Earth's most Mars-like environments, harbor cryptoendolithic microorganisms that live within rock pores and crevices. These communities have evolved remarkable survival strategies, including specialized pigments, that protect against UV radiation and metabolic systems that function at extremely slow rates during harsh winter conditions. High-altitude environments create additional evolutionary pressures that have shaped unique adaptations. The Atacama Desert, located at elevations exceeding 4,000 meters in the Chilean Andes, experiences intense solar radiation, extreme temperature variations, and virtually no precipitation. Microorganisms inhabiting this environment have developed enhanced DNA repair mechanisms and specialized protective compounds that shield against radiation damage. Some species enter dormant states for extended periods, reviving only when rare moisture becomes available. Salt lakes and hypersaline environments have fostered the evolution of halophilic organisms that require high salt concentrations for survival. These environments, such as the Dead Sea and Great Salt Lake, contain salt concentrations up to 10 times higher than seawater. Halophilic archaea have evolved unique cellular mechanisms, including specialized salt-dependent proteins and osmotic regulation systems that maintain cellular integrity in these extreme conditions. Space exposure experiments have revealed that some Earth organisms can survive conditions beyond our planet's protective atmosphere. The European Space Agency's EXPOSE experiments have subjected various organisms to space's vacuum, temperature extremes, and cosmic radiation, Results demonstrate that certain bacterial spores, lichens, and tardigrades can survive extended space exposure, suggesting potential for life transfer between planetary bodies through panspermia. These extreme environment studies have profound implications for astrobiology and the search for extraterrestrial life. Understanding how Earth's extremophiles survive and thrive in harsh conditions informs models of potential life on Mars, Europa, Enceladus, and other celestial bodies with extreme environments. The molecular mechanisms discovered in terrestrial extremophiles provide blueprints for identifying biosignatures and designing life detection experiments for future space missions. The remarkable survival capabilities of Earth's extremophiles have fundamentally transformed astrobiology, expanding the concept of habitability far beyond the traditional Goldilocks zone, where liquid water can exist on planetary surfaces. These microscopic pioneers demonstrate that life can flourish in conditions previously considered absolutely lethal, forcing scientists to reconsider where and how life might exist throughout the cosmos. Traditional models of planetary habitability focused narrowly on surface conditions similar to Earth's temperate regions. The habitable zone, or circumstellar habitable zone, was defined as the orbital distance, where planets could maintain liquid water on their surfaces. However, discoveries of extremophiles thriving in environments resembling conditions on Mars, Europa, and Enceladus have dramatically broadened these parameters. Scientists now recognize that habitable environments may exist far beyond conventional zones, including subsurface oceans on icy moons and the deep interiors of seemingly hostile worlds. Mars presents perhaps the most compelling case for extremophile-informed astrobiology. The red planet's current surface conditions, extreme cold, intense radiation, and an atmosphere primarily composed of carbon dioxide, closely mirror environments where terrestrial extremophiles flourish. Perchlorate salts discovered in Martian soil, once considered prohibitive to life, are now known to support certain halophilic organisms on Earth. The seasonal appearance of recurring slope lineae, potentially indicating transient liquid water, suggests environments where extremophiles might survive. Recent discoveries by NASA's Perseverance rover in Jezero crater have identified organic compounds and minerals indicating past hydrothermal activity. These findings are particularly significant because Earth's hydrothermal vents harbor some of our planet's most extreme life forms. The mineral assemblages detected on Mars, including clays and sulfates formed in aqueous environments, provide potential habitats for chemosynthetic organisms similar to those found in terrestrial extreme environments. Jupiter's moon Europa represents another prime target for extremophile-based life detection strategies. Beneath its icy surface lies an ocean containing more than twice the volume of all Earth's oceans combined. The interface between Europa's rocky core and liquid ocean likely creates hydrothermal activity similar to Earth's deep-sea vents. The discovery of water plumes erupting from Europa's surface suggests that sampling this subsurface environment may be possible without drilling through kilometers of ice. Saturn's moon Enceladus offers equally compelling prospects for extremophile-type life. The Cassini spacecraft detected hydrothermal activity on Enceladus's ocean floor, confirmed by the presence of silica nanoparticles in its ice plumes. These particles indicate water-rock interactions at temperatures between 90 and 200 degrees Celsius, ideal conditions for thermophilic organisms. Additionally, Cassini detected hydrogen in Enceladus's plumes, suggesting serpentinization reactions that could provide energy for chemosynthetic life. The search for biosignatures has been revolutionized by extremophile research. Traditional approaches focused on oxygen and ozone as indicators of photosynthetic life. However, studies of Earth's extremophiles have identified alternative biosignatures, including specific isotopic ratios, unusual mineral formations, and trace gases produced by chemosynthetic processes. Methane detection on Mars and in the atmospheres of exoplanets has gained new significance, as methanogenic archaea represent some of Earth's most successful extremophiles. Advanced missions incorporating extremophile insights are already in development. NASA's Europa Clipper mission, launching in 2024, will search for conditions suitable for extremophile-type life in Europa's subsurface ocean. The mission includes instruments designed to detect organic compounds, analyze ice composition, and characterize the Moon's habitability potential. Similarly, the planned Enceladus Life Finder mission will specifically target biosignature detection. In the Moon's plume material, exoplanet research has also been transformed by extremophile discoveries. Planets orbiting red dwarf stars, once considered uninhabitable due to intense stellar radiation and tidal locking, are now recognized as potential habitats for radiation-resistant extremophiles. The recent discovery of 7 Earth-sized planets in the TRAPPIST-1 system has sparked renewed interest in these environments, particularly given that several planets may harbor subsurface oceans maintained by tidal heating. The implications extend beyond simple life detection to understanding life's cosmic distribution and evolution. If extremophiles can survive space travel, as suggested by experiments with tardigrades and bacterial spores, panspermia, the transfer of life between planets, becomes more plausible. This possibility suggests that life might be more widespread throughout the universe than previously imagined, with extremophiles serving as cosmic seeds capable of establishing new biospheres on suitable worlds. The extraordinary biological mechanisms discovered in extremophiles are rapidly translating into revolutionary applications that promise to transform medicine, industry, and space exploration. These microscopic survivors, forged in Earth's most hostile environments, possess molecular toolkits that offer solutions to some of humanity's most pressing technological challenges, from preserving organs for transplantation to enabling long-duration space missions. Medical applications of extremophile research represent one of the most promising frontiers in biotechnology. Tardigrade-specific, intrinsically disordered proteins are being investigated for their potential to preserve human cells and tissues during medical procedures. Researchers at several institutions are developing TDP-based preservation systems that could revolutionize organ transplantation by extending viable storage times from hours to potentially weeks or months. This breakthrough could dramatically increase organ availability and improve transplant success rates worldwide. Cancer treatment research has also benefited from extremophile studies. The exceptional DNA repair mechanisms found in tardigrades and radiation-resistant bacteria, like Deinococcus radiodurans, are providing insights into protecting healthy cells during radiation therapy. Scientists are investigating how extremophile repair pathways might be engineered into human cells to reduce radiation therapy side effects while maintaining treatment efficacy. Additionally, the study of extremophile stress response systems is informing new approaches to cancer cell targeting and drug resistance mechanisms. Industrial biotechnology has been revolutionized by thermophilic enzyme applications. The discovery of heat-stable enzymes from organisms like Thermus aquaticus launched the modern molecular biology era with the development of polymerase chain reaction technology. TAQ polymerase enables rapid DNA amplification and has applications ranging from medical diagnostics to forensic analysis. The global PCR market, valued at billions of dollars annually, demonstrates the economic impact of extremophile-derived technologies. Manufacturing industries increasingly rely on extremophile enzymes for processes requiring high temperatures or harsh chemical conditions. Thermostable enzymes are used in paper production, where they break down lignin at temperatures exceeding 90 degrees Celsius. Textile manufacturing employs these enzymes for fabric processing and dye removal. Biofuel production utilizes thermophilic cellulases and hemicellulases to break down plant biomass more efficiently than conventional enzymes, contributing to sustainable energy development. Agricultural applications of extremophile research are addressing global food security challenges. Drought-resistant crops are being developed using genes from desert-dwelling extremophiles that can survive extreme desiccation. Scientists have successfully transferred anhydrobiosis-related genes from tardigrades into crop plants, creating varieties that can survive extended periods without water. These developments are particularly crucial as climate change increases drought frequency and severity worldwide. Space exploration applications represent perhaps the most exciting frontier for extremophile biotechnology. NASA and other space agencies are investigating how extremophile survival mechanisms might protect astronauts during long-duration missions to Mars and beyond. Research focuses on developing radiation protection systems based on extremophile DNA repair mechanisms and cellular protection strategies. Additionally, extremophile-derived preservation technologies could enable long-term storage of food, medicine, and biological samples during multi-year space missions. Closed-loop life support systems for space habitats are incorporating extremophile-based biotechnology. Thermophilic organisms are being engineered to process waste materials and produce useful compounds in space environments. These biological systems could reduce mission mass and complexity while providing sustainable resource recycling capabilities. Some extremophiles are being investigated for their potential to produce oxygen, food, and pharmaceuticals using minimal resources available on other planets. Environmental remediation applications leverage extremophile capabilities to address pollution and contamination challenges. Acid-resistant bacteria are being used to neutralize acid mine drainage, while salt-tolerant microorganisms help restore soil in areas affected by salinization. Radiation-resistant organisms show promise for cleaning up radioactive contamination at nuclear accident sites. These bioremediation approaches offer sustainable alternatives to traditional chemical cleanup methods. Synthetic biology approaches are combining extremophile mechanisms with genetic engineering to create novel organisms. With designed capabilities, scientists are developing artificial cells that incorporate multiple extremophile survival strategies, potentially creating organisms capable of surviving in multiple extreme conditions simultaneously. These engineered systems could serve as biological factories for producing valuable compounds under harsh industrial conditions or in space environments. The pharmaceutical industry is exploring extremophile compounds for drug development. Many extremophiles produce unique secondary metabolites with antimicrobial, antiviral, or anti-cancer properties. The harsh environments that shape these organisms often result in the production of novel bioactive compounds not found elsewhere in nature. High-throughput screening of extremophile extracts is identifying promising candidates for drug development, particularly antibiotics effective against drug-resistant pathogens. As we stand at the threshold of unprecedented technological advancement, the study of extremophiles is poised to unlock secrets that will fundamentally reshape our understanding of life's boundaries and possibilities. Cutting-edge research techniques. From advanced genomic sequencing to sophisticated space-based experiments, are revealing new dimensions of extremophile biology that promise to revolutionize multiple scientific disciplines and practical applications. Single-cell genomics represents one of the most transformative developments in extremophile research. Traditional cultivation methods can only access a tiny fraction of microbial diversity, as most extremophiles resist laboratory culture. Advanced techniques now allow scientists to sequence the genomes of individual uncultured microorganisms directly from environmental samples. This approach has revealed thousands of previously unknown extremophile species and uncovered novel metabolic pathways that expand our conception of biochemical possibility. The Human Genome Project's completion in 2003 catalyzed similar ambitious efforts focused on extremophile communities. The Earth Microbiome Project and similar initiatives are systematically cataloging microbial diversity across extreme environments worldwide. These comprehensive surveys are revealing that extremophile diversity far exceeds previous estimates, with each extreme environment harboring unique communities adapted to specific combinations of environmental stresses. Advanced imaging technologies are providing unprecedented insights into extremophile cellular architecture and survival mechanisms. Cryo-electron microscopy allows researchers to visualize cellular structures at near-atomic resolution without the damage caused by traditional preparation methods. These techniques have revealed intricate details of how extremophile proteins maintain their structure under extreme conditions and how cellular membranes adapt to temperature and pressure. Extremes. Synthetic biology approaches are advancing rapidly, enabling researchers to engineer organisms with designed extremophile characteristics. Scientists are developing modular genetic systems that can be combined to create organisms capable of surviving multiple extreme conditions simultaneously. These synthetic extremophiles could serve as biological factories for producing valuable compounds under harsh industrial conditions or as components of life support systems for space exploration. Quantum biology is emerging as a new frontier in extremophile research, investigating whether quantum mechanical effects play roles in extremophile survival mechanisms. Some researchers hypothesize that quantum coherence might contribute to the exceptional efficiency of extremophile DNA repair systems or the stability of their protective proteins. While still speculative, these investigations could reveal entirely new principles underlying life's resilience. Machine learning and artificial intelligence are accelerating extremophile research by identifying patterns in vast datasets that would be impossible for humans to detect. Advanced algorithms can predict which environmental conditions might harbor previously unknown extremophiles and identify promising candidates for biotechnological applications. AI systems are also being used to design improved versions of extremophile enzymes with enhanced stability or novel properties. Space-based research platforms are expanding extremophile studies beyond Earth's limitations. The International Space Station regularly hosts experiments exposing various organisms to space conditions, providing insights into the limits of biological survival. Future platforms, including lunar research stations and Mars habitats, will enable long-term studies of how extremophiles adapt to extraterrestrial environments. The search for life beyond Earth is increasingly informed by extremophile research. Advanced biosignature detection techniques are being developed based on the metabolic products and cellular structures of terrestrial extremophiles. Future missions to Mars, Europa, and Enceladus will carry instruments specifically designed to detect extremophile-type life forms. The discovery of extraterrestrial extremophiles would represent one of the most significant scientific breakthroughs in human history. Climate change research is leveraging extremophile studies to understand how life might adapt to rapidly changing environmental conditions. As global temperatures rise and extreme weather events become more frequent, understanding extremophile adaptation mechanisms provides insights into ecosystem resilience and potential intervention strategies. Some researchers are investigating whether extremophile characteristics could be introduced into endangered species to enhance their survival prospects. Emerging applications in nanotechnology and materials science are drawing inspiration from extremophile survival strategies. The protective mechanisms used by extremophiles are informing the development of new materials that can withstand extreme conditions. These bio-inspired materials have applications ranging from protective coatings for spacecraft to medical implants that must function in harsh biological environments. The philosophical implications of extremophile research extend far beyond scientific applications. These organisms challenge fundamental assumptions about life's requirements and limitations, suggesting that consciousness and complexity might arise under conditions previously thought impossible. As we discover life thriving in increasingly extreme conditions, we must reconsider what it means to be alive and where the boundaries of existence truly lie. The future of extremophile research promises discoveries that will transform our understanding of life itself, opening new possibilities for human survival and expansion beyond Earth, while providing solutions to pressing challenges on our home planet.